Have you ever looked at an exam question and you know the answer, but you're just not sure how to phrase it to get the marks? Well, today, we're going to decode a GCSE biology question to show you how the official mark scheme is basically your secret weapon for getting every single point.
So we're diving into question 3 from an AQA paper. It's a classic required practical, all about preparing a microscope slide with onion cells. This one's a real all-rounder, because it doesn't just test your lab skills, it tests if you get why you're doing each step, and it even throws in some math and drawing. And this is the image that students were given to work with, magnified 400 times. It's the starting point for the whole question, so take a good look. Yeah, recognizing the onion cells is the easy part. Answering the questions perfectly, that's where the real challenge is. So how do you make sure you're giving the examiners exactly what they're looking for? You can forget all the guesswork. The key isn't some secret technique, it's actually a document you can find online. And here it is, the official AQA mark scheme. Now, this thing isn't just an answer key. It's basically the examiner's playbook. So let's walk through question 3 together and see exactly which words and phrases will earn you those top marks. Okay, let's kick things off with the first part of the question. You're asked to complete a risk assessment for the experiment. This is all about thinking like a scientist. What could possibly go wrong here, and what are you going to do to stop it from happening? You know, the first thing you'll notice here is the flexibility. For each hazard, the mark scheme gives you a bunch of different correct answers. This just shows that examiners are looking for practical, common-sense solutions, not just one specific answer you've had to memorize. As long as what you suggest is safe and makes sense, you're going to get the marks. Right. Next up, the exam shifts from safety to the actual scientific reasoning. See, it's not enough to just know the steps of the experiment. You've got to understand why each one is so important. Let's look at the 3 key steps they ask about. Just look at how short and direct these answers are. Examiners want you to get straight to the scientific point. No need for long, waffling explanations. Using those key words like stain to make things visible or identifying air bubbles as the problem, that's what locks in these marks. Okay, now for the bit that a lot of students find kind of tricky, the calculation. And listen, showing your work is absolutely crucial here. The mark scheme literally gives out marks for each correct step, so even if you mess up the final answer, you can still pick up some really valuable points along the way. So, here's the problem they give you. The image of the cells is 4.8 centimeters long, and the magnification is times 400. Your job is to figure out the actual length of the cells in micrometers. The mark scheme breaks this down into such a clear, logical process. You get a mark just for rearranging the magnification formula correctly. Then you get another one for substituting the values in to get 0.012 centimeters. And that final mark, that's for the correct conversion to micrometers. This is exactly why showing your working is so important. And there you have it, our final answer, 120 micrometers. By just following the steps laid out in the mark scheme, you can get there methodically and, most importantly, accurately. We're on the home stretch now. The final part of the question switches gears from calculations over to your observational skills. It's all about scientific drawing and knowing the limits of a light microscope. So, have a look. On the left is the original image, and on the right is a student's drawing. You can probably spot some pretty clear differences right away. The exam wants you to analyze this drawing with a scientific eye and point out how it could be better. The question itself is pretty simple. What are 2 ways to improve this drawing? Think about it for a second. What makes a good scientific drawing? What are the key rules you've been taught in class? The mark scheme basically gives us a perfect checklist for the rules of scientific drawing. Any 2 of these points would get you full marks. For example, did you spot the shading? That's a classic mistake. The lines also have gaps, and some of the cells are drawn overlapping. Other good points would be to add a magnification scale, use solid, continuous lines, or label the different parts of the cell. Knowing these basic rules is so essential. And now for the final question, which tests your broader knowledge. What if we swapped out our regular light microscope for a super powerful electron microscope? How would that change what we see when we look at these onion cells? So the mark scheme is really looking for 2 key ideas here. First, an electron microscope has a much higher magnification, so the image would be way bigger. Second, it's got a higher resolution, which means you'd see much more fine detail. You could also get a mark by saying you'd see more subcellular structures. You know, those tiny organelles inside the cell, like ribosomes and mitochondria, which are totally invisible with a light microscope. It just shows a deeper understanding of the tech. So, we've just broken down a whole 14-mark question using the examiner's own guide.
What's the key takeaway from all this? What's the most important lesson you can apply to any exam you're prepping for?
It's this. The mark scheme is your blueprint for success. It shows you the level of detail you need, the specific words to use, and exactly how the marks are given out. By studying it alongside past papers, you're not just learning the subject, you're learning to speak the language of the exam. And that is how you turn your knowledge into top marks.